Hello, I'm Rachel Karchin, and I'm a professor of biomedical engineering, oncology, and computer science at Johns Hopkins University. Today I'm going to talk to you about how you can customize your variant interpretation workflow with OpenCRAVAT. OpenCRAVAT is an acronym for the Open Custom Ranked Analysis of Variants Toolkit. OpenCRAVAT is an open source platform for analyzing and annotating genetic variants. And it can be used in a variety of case studies, both for individuals, for cohorts of individuals, and even for case control studies. We have over 300 tools available for you to assemble your workflow. The entire platform can be installed very easily, and you'll find that it's thoroughly customizable in terms of every step of your workflow. You'll begin by collecting the samples that you want to analyze and outputting your variant calls for the samples into a standard variant input format. We support many input formats, including VCF, genotype files from 23andMe, Ancestry, family tree DNA, and you can also put in lists of IDs in dbSNP or HGVS format. If you want to use an input format that we don't currently support, you can code up your own input format using a tool that we call an input converter. And we provide detailed instructions of how you can code up your converter. In your next step, you'll go to our annotator store, where we have hundreds of tools available for you to select from. You will then use your selected tools, run them on your variants, and you will be annotating your variants with all the different tools that you've selected. Next, you can do interactive viewing of all the annotations you've run. And for convenience, we have very complex but intuitive filters that you can use. And you can build JSON files for these, or you can use our GUI to build your filters in a visual style. This allows you to narrow down from millions of variants to a few variants that meet the criteria that you're the most interested in. Finally, you can output your results into many formats: VCF, CSV, TSV. You can use a SQLite database format. This is a lightweight database where you can store all your results and also the visualizations you've put together with your interactive viewing. And like with the inputs, if there are annotators that we don't have that you would like, or output reporters to support output formats that you'd like to use, we provide the capability to write your own annotator, to write your own output reporter, and even to post these into our library of tools. This analysis can also be run completely on the command line, which is convenient for running OpenCRAVAT within a much larger pipeline, where perhaps you begin with FASTQ or BAM files and do your variant calling before inputting your variants for annotation, and also where you may want to direct your outputs into additional analysis tools for various genotype-phenotype analysis, fine mapping, or gene burden tests. On this slide, I'm breaking down the steps I've just described, and you can use these QR codes to go to websites that will give you much more detail about all these steps. You can see here all the different input formats that we support. And you can also see on our GUI how you submit your variants by selecting a genome assembly, uploading the file format of your choice, then running your job with selected annotators. Here is a graphical view of our store, and here is a checklist that you can use in the GUI to select the annotators that you want to use. And in the GUI, we also have these annotators broken down by category. So it's easy to find them. They're indexed, you can search them, they're tagged. You can view them under various larger topics, such as cancer-related variants, clinical annotations, and so forth. Here is a view of how you can use filtering in the GUI. You can filter both on the sample level, the gene level, and on the variant level. And you can combine these filters in all sorts of creative ways. While we provide some standard filters for variants, we also have a tool called the Query Builder, which allows you to construct filters using the fields reported by any annotators you've selected in your job. So on this slide, you can see here that I've built a simple filter. I would here like to see variants that have some kind of data associated with them in CIVIC, a clinical annotation database, and I would also like variants that are annotated as likely pathogenic, pathogenic or likely pathogenic, a risk factor, and so forth in the ClinVar database. And I did this with a drop-down menu and a checklist. You can see, applying these filters, I can reduce the number of variants in my job. This was from a single individual whole genome sample down from really a very large number. This was actually several million, down to 22 variants that meet the criteria that I'm interested in here. I can also output in this list of different types of reporters. Again, you can make your own reporter. You can make your own annotators. And finally, you can do all of this on the command line to integrate with other tools, such as variant callers, genotype, phenotype, analysis tools. And I've already told you about that, but you can see on this slide there's just an outline of some of the commands you would use on the command line to set up OpenCRAVAT, to run it, and then to do additional analysis. And I also have a shot here of a piece of what the annotator store looks like on the command line. So anything you could do in the GUI, you can also do in the command line. I think you'll find that OpenCRAVAT is very flexible. You'll be surprised at how easy it is to install and work with when compared to some other comparable tools, which I won't name right here. So thank you very much for taking the time to listen to my talk. And please feel free to reach out to talk to me. And there is a very nice way to do this on the ISMB website. Thanks so much.